The cardiac myocyte. Like all other tissues, the heart muscle is formed of unit cells, called the myocytes. These are the contractile muscle fibers of the heart. The length of the cardiac myocyte is 100-150 micrometer, and its width is 10-20 micrometer. Histologically, the fiber is formed of smaller fibrils, which in turn consist of tiny filaments. This is a schematic cross-section of the myocyte, muscle cell, or muscle fiber, showing the cell membrane, muscle fibrils, and filaments. Two types of filaments, thick and thin, are identified. The thick filaments are composed of one type of protein called myosin, while the thin filaments are composed of three types of proteins, actin, troponin and tropomyosin. On microscopy, a fibril is seen divided into segments called the sarcomeres. In the middle of each sarcomere a dark band, anisotropic or A band, is recognized, while on either side of the A band, a light band, isotropic or I band, can be seen. The A band is dark because it contains the thick and thin filaments, while the lighter I bands contain only the thin filaments. The thick filaments extend all along the A band, while the thin filaments extend from both sides toward the center of the A band, without meeting, but leaving a pale zone in the middle called the H zone. In the middle of the H zone, however, there is another dense line, M line or midline, formed by condensed proteins. This figure shows the sarcomeres, each composed of an A band in the middle, and I bands on either side. The I bands of two sarcomeres thus come nearby, separated by the Z line, which is dark because of overlap of thin filaments from both sarcomeres. The thin filaments in an I band of one sarcomere overlap with those in the neighbor sarcomere, to form a dense line, Z line, or end line. As such, we can conclude that a sarcomere is a segment of a myofibril, extending between two Z lines. Here is one sarcomere, with the A band in the middle, and I bands on either side, and shown also are the H zone, M line, myosin thick filaments and actin thin filaments. The thick myosin filaments are connected to the Z lines on either side of the sarcomere by another type of protein called titin. Another important structure within the myocyte is the T-transverse tubule, which runs from the plasma membrane, deeply into the interior of the cell, allowing quick penetration of the depolarization wave. In this way, excitation-contraction coupling becomes feasible. Although it is made up of discrete cells, the heart muscle functions as a syncytium, thanks to the extensive branching and interdigitization, providing the substrate for both mechanical and electrical cohesion. Between myocytes, special structures are present, called the intercalated discs, which contain low-resistance pathways called the gap junctions, allowing rapid conduction of the electrical current, ensuring that the heart muscle is working as a syncytium. Troponin and tropomyosin represent regulatory proteins, controlled by the concentration of intracellular calcium. Calcium is released from the sarcoplasmic reticulum on depolarization, to initiate contraction, then reabsorbed to terminate contraction. Troponin is a protein complex, formed of three subunits. T subunit, tropomyosin binding. C subunit, calcium binding, and I subunit, inhibitory troponin, which inhibits the activation of myosin adenosine triphosphatase by actin. It is present only in cardiac and skeletal muscles, not in other tissues. Troponin and tropomyosin prevent the contact between actin and myosin at rest. Upon stimulation, calcium is released and binds to troponin C subunit. Calcium-bound troponin C subunit induces a conformational change of tropomyosin. Modification of tropomyosin allows actin-myosin interaction. The three subunits of troponin, T, C, and I, connect actin to tropomyosin in the resting state, while tropomyosin covers myosin binding sites on actin filaments, preventing contraction. Upon stimulation, calcium released from the endoplasmic reticulum binds to the C subunit, detaching the I subunit and tropomyosin from actin. Myosin binding sites are uncovered, and adenosine triphosphatase is stimulated, producing cyclic adenosine monophosphate and energy for myosin to bind to actin, resulting in muscle contraction. Troponin subunits have different isoforms in cardiac and skeletal muscles. There are several cardiac-specific isoforms, and skeletal muscle-specific isoforms, of the T subunit and the I subunit of troponin. On the other hand, the C subunits are nonspecific. In our cardiovascular clinical practice, we test the isoforms of the cardiac-specific troponin subunits. Cardiac-specific troponin T, CTNT, or cardiac-specific troponin I, TNI. Consequently, a positive test always means myocardial injury. It is imperative to realize that myocardial injury, and consequently elevation of cardiac-specific troponins, may be due to both cardiac and non-cardiac diseases. Cardiac causes of elevated cardiac troponins. Myocardial infarction, prolonged coronary vasospasm, percutaneous or surgical coronary intervention. Myocarditis. All forms of cardiomyopathy, dilated, hypertrophic, restrictive, peripartum and Tacot subu. Left ventricular hypertrophy. Heart failure. Severe tachyarrhythmia. Cardiac contusion, DC shock, cardiac surgery, heart transplantation, radiofrequency ablation. Non-cardiac causes exert indirect effects on the heart muscle. Aortic dissection may result in acute myocardial infarction, and acute left ventricular pressure overload. Acute left ventricular pressure overload can also occur with hypertensive crisis. Right ventricular pressure overload may be due to primary pulmonary hypertension, pulmonary embolism, or exacerbation of chronic obstructive pulmonary disease. Toxic myocarditis may be due to renal failure, drugs, toxins, sepsis, or poisoning. Cardiac toxicity from sympathetic overactivity may occur in strenuous exercise, and in central nervous system diseases such as strokes, subdural hemorrhage, and seizures. Skeletal muscle diseases may also affect the heart muscle, such as polymyositis, dermatomyositis, and rhabdomyolysis. Cardiac metabolism is almost exclusively aerobic. Heart muscle does not tolerate lack of oxygen well. Mitochondria provide the energy for the huge number of heartbeats that will occur during a human lifespan. Normally, energy is provided to heart muscle by fat, 60% of energy. Half of this energy comes from circulating free fatty acids. Carbohydrates, 35%, and ketones, 5%. There are abundant capillaries through the myocardium, and a large amount of myoglobin within the heart muscle, ensuring appropriate blood, nutrients, and oxygen supply. Adenosine triphosphate, ADP, is hydrolyzed by actin utilizing the adenosine triphosphatase enzyme on the myosin head. The tiny head of a myosin molecule swivels, dragging itself and ultimately the whole myocardium, along the actin railway track. The cardiac muscle possesses some additional properties over the skeletal and the smooth muscles, namely, morphological, electrical, mechanical, metabolic, and special properties. The cardiac muscle is characterized by the following morphological properties: striations due to regular arrangement of thin and thick filaments, branching and interdigitization of muscle fibers, and the intercalated discs and gap junctions, which allow functional syncytium. Electrically, in the resting polarized condition, the membrane potential in the cardiac muscle fiber is around -90 mV. On stimulation, the action potential has a characteristic plateau phase which delays the completion of repolarization phase and thus prolongs the effective refractory period of the cardiac muscle. Mechanically, the cardiac muscle cannot be tetanized, unlike the skeletal muscle, because it cannot be stimulated before it completes its contraction phase, thanks to its longer refractory period, and the characteristic plateau phase of the action potential, which delays the completion of repolarization. From the metabolic point of view, large number of mitochondria and high myoglobin content ensure maximum oxygen supply and aerobic respiration. Cardiac muscle, however, can also utilize lactic acid, a characteristic property that reduces the chance of getting fatigue. Special properties include automaticity or autorhythmicity, excitability, conductivity, contractility, and lucitropy. Automaticity or autorhythmicity. The automatic cell can generate action potentials without any external stimuli, thus acting as a pacemaker for the cardiac muscle, thanks to its continuous automatic slowly depolarizing membrane potential during diastole, instead of a steady resting membrane potential. Influencing the automaticity of the muscle is called chronotropism. Excitability is the ability of a cell to respond and develop an action potential, in the presence of a sufficient stimulus or local current. Influencing the excitability of muscles is called bathmotropism. Conductivity is the traveling of the impulse along the tissue. Conduction velocity of the cardiac muscle is about 1 meter per second, while the cardiac conduction system conducts relatively faster. Influencing the conductivity is called dromotropism. Contractility is the ability of shortening, in reaction to an appropriate membrane depolarization. Influencing the contractility of the muscle is called inotropism. Lucitropy is the rate of relaxation of the cardiac muscle. Influencing the relaxation of muscle is called lucitropism. Thank you.